Hi everyone, welcome back. This week in Module 3, we're going to be focusing in on the end of Chapter 2, where we're going to pick up from where we left off looking at proteins and nucleic acids, and we're going to look at those last two macromolecules, the carbohydrates and the lipids. We'll then move on to taking a look at the beginning of Chapter 3, where we're going to start to begin to look at cells, looking at their structure and then the functions of many of their organelles. Now, we're going to start off with carbohydrates today. Now, carbs make up the structural framework of cells and play a critical role in both energy production as well as storage. We are going to spend an entire week in about, I believe it's 2 weeks, where we are going to focus on cellular respiration, which is the process by which cells take in glucose. And then they put it through glycolysis and then in the presence of oxygen to then produce usable energy in the form of ATP. All right, we're also then going to talk about what happens when you have excess glucose in your diet, right? We have an ability to be able to take that excess glucose and then store it up as polysaccharides known as glycogen within our liver as well as within our muscle tissue. Now, carbohydrate defined as any molecule containing the elements C, H, and O. So quick review, what are the 4 atoms that you can expect to find within any of the organic macromolecules? So you've got carbon, you've got hydrogen, you've got oxygen. What was that last one? Nitrogen, right? We looked at the 2 macromolecules last week that contain nitrogen. This week, we're going to be focusing on both carbs and fats. And carbs and fats do not contain nitrogen. You might be thinking, why does that matter? It matters because our body is regularly going to be wanting to break down carbohydrates and break down fats to produce energy. Well, if we do that, all of our byproducts end up being carbon dioxide and water. Okay, a relatively clean burn. Now, if we start to break down proteins and nucleic acids that we looked at last week, that's the last resort for our body to want to try to break down a macromolecule to obtain energy because we're left with nitrogenous wastes. And to get rid of it, we need to convert that to ammonia and then in the liver, convert that to urea, which we then secrete from the body through our urine. All right, so it's a very complicated process, and our body then has to do something with the nitrogenous waste that's left over. So our body's first macromolecule that it's going to target to break down to produce energy is going to be your carbohydrates. Okay, second would be the fats, and then last resort sort of is the proteins. Now, one of the key things here with carbohydrates is that carbohydrates are always going to exist with the ratio of 1 carbon to 2 hydrogens to 1 oxygen. And you're going to see that repeated as we look at the different molecular structures of all of the monomers and the disaccharides, monosaccharides and disaccharides. Glucose, for example, is C6H12O6. Right? So following that 1 to 2 to 1 ratio. We have 2 main classes of carbohydrates. We have simple carbs and we have complex carbohydrates. All right, now simple carbohydrates, this is where glucose fits in, but also lactose, other disaccharides as well. So we have 2 classes, those that are made up of 1 monomer and those that are made up of 2 monomers. All right, the single monomers we refer to as monosaccharides. Those that are made up of 2 monomers we refer to as disaccharides. Right, now the term saccharide, remember how we talked about how everything comes from Greek and Latin prefixes and suffixes? Well, saccharide originates from a Greek term for sweet. Monosaccharide, single sugar, disaccharide, 2 sugars. Now our complex carbohydrates, these are the polymers. And we refer to these as polysaccharides. So polysaccharide, by definition, is any carbohydrate that is made up of more than 2 monomers. Okay, and typically we see these, for example, glycogen or starch or cellulose. Those all occur in big long chains. All right. Remember how I said last week that's really important that you understand and remember the structure of these different monomers? With carbohydrates, remember it's CHO, and it's going to always have a ring structure to it. Okay, so all of your carbohydrates are always going to hold this ring structure. So if you have 2 monosaccharides bound together, you know it's a disaccharide, right? So 2 ring structures bound together. When you compare this to the fatty acid chains, they look completely different. And when you compare them then to nucleic acids as well as to proteins, looking at the amino acids, those 2 contain nitrogen within them. All right, so by the time you then have these 4 different monomers side by side, making up the 4 different macromolecules, I'm hoping that you're going to find it pretty easy to be able to identify which is which. So in this case here, we're going to start with monosaccharides, right? So consisting of only one monomer subunit. So the example that we're going to start with is looking at our simple sugar, glucose. So glucose is the monosaccharide that we are able to use to break down and make ATP. Fructose occurs within fruits. It's what gives it the sort of the sweetness. But fructose actually is processed in the liver and is converted to fat. Okay, so fructose doesn't go through the same process to produce energy in the way that glucose does. And this is where we run into a problem with foods that are made up of high fructose corn syrup. And we'll talk about that in a little bit. Okay, so glucose and fructose are both monosaccharides. And then we have galactose. Okay, so you might be thinking, hey, I've heard of lactose before. Okay, well, lactose is a disaccharide and is actually partly made up of galactose. All right, so each of these are monosaccharides. These are the basic simple sugars. And now when we combine these together with glucose, we then make disaccharides. Now, high fructose corn syrup is found in many different processed foods. Fructose is the sweetest of all of the sugars. And with high fructose corn syrup added, the problem with that is they're getting a lot of sweetness, and it's really cheap for companies to be able to add that sweetness without having to produce other sweeteners, okay, because corn is so readily available. Now, as I mentioned before, fructose is processed by the liver. And so here it's converted directly into fat, and it's not used by our cells to produce energy the way that glucose is. And so right away, the more of this fructose that you're taking in, you're going to be converting all of the fructose in that into fat directly. All right, and so where is it found? Oh no, let's not look at this. No, okay, I will. Cereals, granola bars, your pops, your energy drinks, your pastries, your cookies, your candies, your sauces, fast food, hamburgers. All right, so apparently we can't eat anything. Okay, that's not the case. It's all in moderation, right? That's what matters. I'm looking at this list thinking like, oh dear, I'm in trouble. Now with high fructose corn syrup, it's roughly a split of 55% fructose and 45% glucose. So when you're, drinking a pop, you still are getting glucose, but there's a huge part of all of that sugar that's in there that is fructose that will be converted into fat. Now, as I mentioned before, your disaccharides, they consist of 2 monosaccharides, right? So we know our monosaccharides are glucose, fructose, and galactose. So the way that our disaccharides are made is that you take a glucose monomer and then you add one of the monosaccharides to it. And you'll see here we have glucose and fructose, glucose and galactose, and glucose and glucose. So let's start here at the top. So first off, we're looking at glucose and fructose, and that is what makes the disaccharide sucrose. Now, sucrose is your table sugar. So yes, even in your processed table sugar, you have fructose in about a 50-50 split with glucose. Okay. Now with lactose, lactose is made from glucose and galactose, and this is the disaccharide that is found within milk products. And then lastly, maltose is the disaccharide that's made up of 2 glucose monomers, and this is what you find in your processed grains. All right, so first off, I want to make a bit of an application here and trip down memory lane to last week when we looked at the process by which we take 2 monomers and we link them together to form new covalent bonds and form a larger polymer. I want you to think, what reaction is taking 2 smaller subunits and putting them together to make the larger molecule? Right? That would be your dehydration synthesis reaction. So you remove the water and you end up with the disaccharide being formed. Now, when we take in something like lactose, we need a specific enzyme. Remember we talked about this? We need enzymes to be able to catalyze these reactions, whether it's putting them together or taking them apart. So with lactose, lactose is a disaccharide in milk. You drink some milk, once it gets to your small intestine, you can't just simply absorb the lactose as is. So what you need is the lactase enzyme in order to break those bonds through a hydrolysis reaction into its individual monosaccharides. Okay, so when this happens, you end up then with glucose and your galactose. We have individuals that are lactose intolerant. Depending on your level of intolerance, it may be referred to just as lactose, sensitivity. But essentially, lactose intolerance is due to the fact that you are lacking the lactase enzyme within your small intestine. So when you drink milk and then you experience the bloating or you experience diarrhea or any kind of stomach pain even after drinking milk, it's likely that you could have some sort of lactose intolerance. And so what you can do is you can take lactase in pill form. It's Lactaid, right? And what Lactaid is it's essentially just the lactase enzyme that you can take orally before you have any kind of dairy products. And then that provides you with the enzyme that you need to break up the lactose. What happens is, though, if that lactose sits around in the intestine, we have lots of bacteria that hang out in there that are part of our normal flora. And what happens is the bacteria in the bowel end up consuming this lactose that was left over, and then they produce gas as a result of eating that sugar. Now, polysaccharides, then, remember, by definition, we're looking at more than two monosaccharides linked together. All right, so at least three. And we can see here an example of a 3 polysaccharide chain creating your starch. We have here linked up glucose units to make our glycogen. Remember, glycogen, we store up glucose in excess in the liver as well as in the muscle tissue so that it's readily available so that when we need it, we can then break down that glycogen into individual glucose units and then use that glucose to create energy. And then also within plants, we can obtain the polysaccharide cellulose. All right, so with the complex carbohydrates, they're long polymer chains of monosaccharides. They contain many carbon-hydrogen bonds and therefore are good for storing energy, right? Those bonds are covalent bonds, which when broken can help to release energy. Now, these bonds are often the ones that are most often broken by organisms to obtain energy, right? This is how any organism is going to obtain their energy. It all comes down to the simple monosaccharide of glucose. All right, so with the carbohydrates, you have the monosaccharides made up of one sugar. You have glucose, fructose, and galactose. You have the disaccharides made up of two monosaccharides. And we had sucrose, which was glucose and fructose, lactose, which was glucose and galactose, and maltose, which was glucose and glucose. And then we had our 3 polysaccharides, our glycogen, our starch, and our cellulose. So we're going to move on now to the last macromolecule and take a look here at the lipids. Okay, so remember, we've already talked about a few of these examples, actually. We briefly I think it was last week, yeah. We took a look at this amphipathic molecule, the phospholipid. Remember, amphipathic refers to the fact that there are 2 regions, a polar region and a non-polar region. We're going to take a look then at these 3 examples, these 3 types of lipids in more detail as we move on throughout the lecture here. All right, so just for now, know that you have the triglycerides, cholesterol, and phospholipids. Lipids play a huge role throughout our body. There's a table in your textbook on page 41, table 2.5. I'll put an example of that as well online if you don't have the textbook. But it just gives you images to pair with all of these individual points just to help organize and also give you a visual for that. Okay? Now, the brain is highly composed of fat. Fat makes up about 60% of the brain and is essential to its functioning. So everything that the brain does, including learning, memory, your mood. Cells, we're going to take a look at those later on today, where we look at how specifically the phospholipids are responsible for making up the cell membranes. Okay, and also within the cell membrane, the phospholipid bilayer, we have cholesterol molecules that exist to help to improve flexibility within the membrane as well. 60% of the heart's energy comes just from burning fats alone. And when you think about what the heart has to do on a daily basis, never stopping, does it ever get a break? No, right? Think about that for a second. It's always going. So 60% of the energy required by each of those cardiac muscle cells is coming from the burning of fat. With nerves, take a look at the myelinated nerves that are located within the textbook. We're going to go through this in a lot more detail in the nervous system lecture. There's insulation that's added to the axons of neurons. And this insulation is acting to provide a way that we can transmit a signal along the length of a neuron down a long passage, say from the spinal cord all the way out on your arm to a muscle without losing that transmission. All right. And so that insulation is known as myelin. The process by which we insulate those neurons is myelination. The lungs, we're going to get to look at the respiratory system next semester. We're going to talk about surfactant in a lot of detail. I think you actually might touch on that in physics a little bit when you look at surface tension. But the lungs produce surfactant, and surfactant is a substance, a fatty substance, that essentially lubricates the insides of the alveoli. Remember the alveoli? That's the structures that are made up of simple squamous epithelium in the lungs. I want you to picture a produce bag and try to open that in the dead of winter when your fingers are dry and everything's staticky and you can't actually get it. You can picture that, right? And you try to get a little bit of mist maybe from the produce section so that you can get your fingers wet and actually open that produce bag. All right, so now that you're all frustrated, it's similar to how surfactant works to be able to keep our lungs, those alveoli, expanding. All right, if we did not have surfactant coating the inside of our alveoli, when we breathe out, those alveoli would just collapse and they would just want to stick together like those staticky produce bags. And so surfactant helps to reduce that surface tension and enables the lungs to be able to stay inflated properly so we can breathe in and breathe out without any collapse. Now, within digestion, fats play a huge role. Fats in a meal are slowing down the digestive process so that you have more time to absorb nutrients and help to provide a constant level of energy and keep the body satiated. So essentially think of this as just satisfied, okay? If you just drink something like, if you just drink fruit juice and you're not taking in any of the bulk from that fruit, your body's not actually going to feel satisfied, right? High fructose corn syrup works in the same way. When you drink anything that has that high fructose corn syrup, it does not satiate you for a long period of time. Whereas fat in a meal, it does. Okay, there's a hormonal cascade that's involved, and we'll talk about that when we look at digestion in biology later on next semester. But we talked about the fat-soluble vitamins last week, right? We looked at A, D, E, and K, and they can only be absorbed if fat is present in the diet. So this is actually something that we didn't talk about when we talked about these last week. If you are taking a multivitamin, you always have to make sure that you are eating some sort of fat in that meal that you're taking it with. Okay, it always needs to be taken with a meal. That's because if you just take it on its own, the water-soluble B and C and a lot of the minerals, they might be absorbed into your body, but your vitamins A, D, E, and K, they're just going to end up staying in your gut and ending up passing through your intestines and exiting your body in your fecal matter. Okay, so you're essentially just literally putting money down the drain in that case. So always make sure that you're taking your multivitamins with some sort of fat in the meal that you're taking them with. That way you can ensure that your fat-soluble vitamins are all being absorbed. Now, organs, there's a great diagram in the text as well of the greater omentum, which is like the apron that covers all of the abdominal organs. We have fat surrounding all of the organs in our body, and we took a look at those different cavities last week. And each of those cavities contains the organs, and there is visceral fat surrounding all of those organs. And that's great because it helps to cushion and it helps to protect the internal organs, right? It's kind of like a nice pillow. The problem becomes, and you can probably already see this coming, right? If we have a cavity that has limited space in it already with the organs that are there, and we start adding more visceral fat into that cavity, we're going to start to eventually squeeze those organs. And this can play a huge role, especially with heart health. If you start increasing the amount of visceral fat that surrounds your heart in its mediastinal cavity, that ends up having a big impact on how hard the heart's going to have to work going forward to be able to contract the same way and pump the same amount of blood as it would if you had less fat. All right, so the heart having to work harder, it's going to have to either beat more or contract more, and there's lots of conditions that can result from that. And then lastly, with the immune system, fats, specifically your fish oils, right, your omega-3s, they help to act as anti-inflammatory fats. So they're essential in your diet. Okay, there are other ones that are pro-inflammatory. There are other fats that are pro-inflammatory, and so you just have to watch out for what it is that you're taking in. But fats in general act to decrease the inflammatory process, and then ultimately, by decreasing inflammation, you're helping your body, your immune system, be able to function as optimally as possible. All right, so taking a look here at the first type of lipid that we're going to talk about, so triglycerides. Now, triglycerides, this is the form that typically we are converting any of the fatty acids we've taken in. We're going to be converting them into triglycerides in order to store them in our adipose tissue. And now the structure of a triglyceride, and it's all in the name actually, comes from the fact that there's a glycerol backbone attached to 3 fatty acid chains. Remember how I said fatty acid chains are just, they're gonna be so obvious, right? It's literally a hydrocarbon chain. It's a long chain of carbons and hydrogens. So your triglycerides are made up of 3 fatty acids. That's where tri comes from. And glyceride comes from the fact that they are all attached to a glycerol backbone. When you're counting your carbons in a fatty acid chain, you want to always start opposite the carboxyl group. Okay, we've already seen a carboxyl group, right? We saw this with the amino acids. That's where they get their term acid from. There's a carboxylic acid attached to the end of them. So you always want to start at this end. And this comes into play when you're starting to count where the double bonds might exist in our unsaturated fatty acids. All right, looking here, we have all single bonds between these carbons, and that would mean that these are all saturated fatty acids. So we have 2 different types. We have saturated and unsaturated fatty acids. And so the difference here is with saturated fatty acids, there are only single bonds that exist between the carbons, and so each carbon is fully saturated with hydrogens. Now, what this means is that you can tightly pack all of these fatty acid chains in together. So this is how that triglyceride would work. If you're looking at a saturated fatty acid in this case, these can pack in nice and tightly. And so at room temperature, they actually remain in their solid form. So if you're ever wondering whether something is saturated or unsaturated fat, you just need to look at what state is it in at room temperature. You do know that if you heat up something like butter or if you heat up something like coconut oil, they will melt, right? But that's because you're changing the configuration due to the increase in temperature. What you need to look at is how does that specific fat exist at room temperature? And that's going to tell you whether it's a saturated or an unsaturated fat. So moving here to the unsaturated fats have double bonds, which means that they are not saturated with hydrogens. And you're going to learn more about this in chemistry. But for example, your carbons are going to have 4 bonds for hydrogens or other atoms that can bond to them. And so in this case, because you have a double bond existing between your carbons, they then do not have any other electrons available to bind with additional hydrogens. All right, so in this case here, with the unsaturated fats, because they have these spaces in their fatty acid tail, they end up actually sort of kinking up like this, sort of bending, all right, in 3D structure. And so by the time you have 3 separate fatty acids that are all gonna, have different double bonds in different places, they're gonna take up a lot of more space, so you can't pack them as tightly together. And that's why they exist in liquid form at room temperature. All right, so typically saturated fats solid at room temperature and your unsaturated fats liquid at room temperature. Now, as I mentioned before, omega-3, 6, and 9 fatty acids, these you get from your diet. They are all essential fatty acids. The name comes from the fact that the omega-3s have their double bond at the 3rd carbon. The omega-6s have a double bond at the 6th carbon, and the omega-9s have their first double bond at the 9 carbon. So we'll take a look at that. So as I mentioned before, omega-3s, great to take on their own because they are anti-inflammatory. You can also obtain your essential fatty acids from fish primarily, But also for those who are vegetarians, you can also obtain them from dark green leafy vegetables as well as nuts and flax seeds. I do know that they do have omega-3 supplements where the origin is plant-based as opposed to fish oils. So now that we've looked at the differences here between saturated versus unsaturated fats, and how to identify them at room temperature, let's take a look at some examples of foods that contain saturated fat versus those that contain unsaturated fat. With saturated fats, you find this in higher proportions within meat, poultry. Chicken actually is quite fatty. Dairy products, as well as eggs, coconut oil, palm oil, butter, and avocados. And then foods that contain unsaturated fats, primarily your oils. So we have olive oil, peanut oil, corn oil, vegetable oil, canola, and safflower oil. All right, so let's look at the difference between cis fatty acids and trans fatty acids. This is known as the cis configuration, and this is known as the trans configuration. Okay, so cis configuration, the hydrogens are on the same side of the fatty acid chain, whereas with the trans configuration, they're on opposite sides of the fatty acid chain. Now, so with the cis fatty acids, okay, this is an example here of an unsaturated fatty acid, right? A monounsaturated in this case, but it's in its cis configuration. Now, what's going to happen with this fatty acid tail is that in 3 dimensions, it's actually going to bend at that double bond, right? So it's going to take up more space. You form a triglyceride made of unsaturated fatty acids. That's going to end up existing as an oil at room temperature. Now, when we fry foods, so at extremely high temperatures, what can happen is that the trans configuration ends up developing, which then turns the originally liquid oils at room temperature into now essentially solids. And so the trans fats also play a huge role in increasing our low density lipoprotein, which is responsible for transporting fat to the bloodstream. So what happens is after you have a meal that's high in trans fatty acids, you end up having a spike in your cholesterol levels. And your LDL is known as your bad cholesterol. By eating more trans fats and sugar, you can actually increase cholesterol levels more than simply by eating more cholesterol. Okay, and what happens is with trans fats as well as sugar, they cause the liver to produce more LDL that contains cholesterol and other fats, so that the trans fats are going to then be taking the fat to the blood vessels. And then in the blood vessels, if your LDL hangs out around there for too long, the cholesterol that it's carrying is going to end up then forming plaque within the blood vessel, which is known as atherosclerosis. Now, another thing that they do is that they take other oils that under, natural circumstances are relatively healthy for us, and they put it through a process known as hydrogenation. Okay, so they put it in the presence of hydrogen gas so that they actually add hydrogens to those double bonds in unsaturated fats. And partially hydrogenating different oils actually makes them more stable, and then it gives them longer storage life. Does this sound like they're doing it to benefit you or benefit them? Now, cholesterol is a very important molecule. Not only is cholesterol found in the cell membranes of all plants, animals, fungi, to add for flexibility within the cell membrane, but it also acts as a precursor to our steroid hormones. And steroid hormones are the fat-soluble hormones that we're going to talk about when we look at the endocrine system. Now, most of the cholesterol in the body is made in the liver. And as I mentioned, if you have a spike in your LDL levels, the LDL is going to transport that cholesterol to your blood vessels and then cause, or be more likely to cause, plaque buildup within those vessel walls. Another thing that cholesterol is extremely useful for is the production of bile. Now, bile is made in the liver and then is stored in the gallbladder. And we use bile then, we secrete it into our small intestine to help to mechanically emulsify fat. So if you want to know what emulsification looks like, take some oil, like just some vegetable oil or something, into a dish, and then put some soap into that. Even if you just put drops of soap into it. What's going to happen is that soap is going to start to break up that oil into little smaller droplets. Okay, and that's the same thing that bile does. It helps to break down the fat mechanically into smaller droplets, which increases the surface area and helps so that when our lipase is going to work on breaking down the fatty acids, it has more surface area to do so. And then cholesterol also, we have our HDL, which is our high density lipoprotein, our good cholesterol, and our LDL, which is our low density lipoprotein, which is our bad cholesterol. All right, so HDL, okay, it helps bring cholesterol back, and LDL takes it to the bloodstream. All right, so LDL, if your numbers are too high, you're more at risk to developing atherosclerotic plaques and make yourself more prone to having blood clots. And then last, but definitely not least, the phospholipids. Now, the phospholipids are the primary fats that are found in cell membranes. Okay, so we're going to take a look at the cell membrane, the structure of it. It forms a phospholipid bilayer where the polar head is oriented towards the extracellular environment and the intracellular environment, because both of those environments are full of water. Now, the fatty acid tails, these are the hydrophobic parts of the phospholipid, and they orient themselves in towards the internal aspect of the cell membrane. So the cell membrane itself is actually a hydrophobic region. All right, remember the term to describe the phospholipid is amphipathic, amphi meaning both, right? So it has both hydrophobic as well as hydrophilic regions. And so we're going to be going from looking at the molecule like this to then seeing how it's put together, and this is how we're going to look at the molecule going forward when we see how it's incorporated in the cell membrane. All right, so here we go. So this is our blown out picture here of our phospholipids making that cell membrane. So as I mentioned before, here we have the cell membrane. It's a double layer known as a phospholipid bilayer. The tails are oriented in towards the inside part of the membrane. All right, so this whole internal portion is a hydrophobic region. Okay, on the intracellular part of the cell membrane, we have the phosphate heads, right? And those are polar, so they're hydrophilic. So they're oriented towards the intracellular portion as well as the extracellular portion. And then space between the phospholipids to add for flexibility, we have the cholesterol molecules. This ends our discussion for this week, looking at the macromolecules. We're then going to move into looking at the structure as well as the function of our cells. By now, we have taken a look at the chemical level of organization in the human body. We've looked at the atoms and how they're put together to make molecules, and then how we take those molecules and through dehydration and synthesis reactions, we make macromolecules, right? So now what we're doing is we're stepping up that pyramid, moving from the chemical level to the cellular level of organization. Now in bio here, we're taking that step up, but we're actually going to be taking a step back from where we were last week in anatomy, right? Because we've already looked at the tissues and the specialized cells that they're made of. So the goal today is to just look generally at cells. We're going to look at the typical eukaryotic cell and what its main structural components are, and as well as the function of those components to the overall function of the cell. Now, as we know, all living things are composed of cells, right? We've looked at the 6 kingdoms, and we've looked at the difference between prokaryotic and eukaryotic cells. We've looked at the difference between unicellular and multicellular organisms. That is one of the key elements to cell theory, which we're going to go through in a few slides ahead. Now, within the human body, the maximum cell size is about 100 micrometers. Now, that happens to be the female egg cell that's the largest. And in contrast, the spermatozoa happens to be the smallest cell that we have in the human body. And so it's interesting that both the largest and the smallest cells are what come together and are united to be able to fertilize that egg and create that diploid zygote. Now, one of the reasons that we have this cap on cell size is that oxygen can only diffuse so far. Okay, so 100 micrometers is about that furthest distance that oxygen can simply diffuse, so move into the cell to be used by it from the external environment. Now, most human cells are roughly between, to give you a reference point, 5 and 20 micrometers. Okay, so the largest here at 100 micrometers, significantly larger than our average. But to give you a broader context of this size, the head of a pin is approximately 2,000 micrometers. So if you were to compare that, the head on a pin is approximately 20 times larger than the ovum, and then more than about 100 times larger than most of our cells, whether it be the epithelial cells, your blood cells, and so on. Cells were first seen for the first time when Robert Hooke developed the microscopes sometime in the 1600s. And then through the years, we've been able to study cells more and more. But this is why we require, 100 times magnification on those microscopes to be able to zoom in enough to be able to visualize these cellular structures. Now, some cells, because we're going to get talking about, different neurons in the body that extend all the way from the spinal cord and run all the way to your muscles that they need to innervate. Those motor neurons are very long. Now, these cells are larger because they have special modifications to increase the surface area. Okay, so first example we're talking about are the neurons. The axons that extend all the way from the cell body, that would be considered an extension, okay, which runs all the way down to the muscle. And then also we've talked about the microvilli on the non-ciliated simple columnar epithelium. Microvilli are those finger-like projections that increase the surface area in order to increase absorption in our small intestine. All right, so there are some modifications to our specialized cells that allow them to kind of go beyond that maximal cell size that we just talked about. But the cell, from the cell membrane to the nucleus and all of the structures within, that structure, the largest would be around 100 micrometers. Now, for organisms to become larger, they have to become multicellular, right? They have to become more complicated. As complicated as your unicellular organisms get, you'd be looking at the protists, right, which are unicellular, made up of one cell, but they are eukaryotic, so they have lots of working parts. Now, as I mentioned, cell theory, these are the basic principles upon which all of our understanding of cells are based. Okay, so first principle is that all living organisms are composed of cells. This is what, leaves out the viruses. Okay, so those are considered non-living organisms for a number of reasons that we've already talked about. Okay, but they are not composed of individual cells with all the working parts. The cell is the basic unit of all living organisms and the smallest single unit that can survive independently. So this is important because we talked about the definition of the cell being the smallest structural and functional unit of life. Also, all cells come from pre-existing cells, right? When you think about sexual reproduction, it involves the sperm, which is haploid, 23 chromosomes, and the egg, which is haploid, 23 chromosomes. When they come together, the sperm fertilizes the egg and you have a diploid zygote. And from that point, all of those cells go through cell division to then continue to multiply to create ultimately the embryological structures and then finally the fully grown fetus. And similar to as well with any of the cells, like we talked about skin this week in anatomy, and the skin cells are always undergoing cellular division, right? The stratum basale keratinocytes are always dividing and producing new cells, okay? And those are coming from the pre-existing cells that already exist in that layer. Another principle is that all cells contain genetic material, the DNA for that cell. And that's something that's unique about every single cell in your body is going to have, if you're reproducing an epithelial cell through cellular division, then you're going to be producing another identical copy. The only cells in our body that are genetically diverse and that we create genetically variable copies of are the gametes, those sex cells. And then lastly, all cells are either prokaryotic or eukaryotic. Okay, remember these two types of cells, prokaryotic are simple, eukaryotic much more complex. So we're going to take a look now at a lot of the cell structures of a generic cell, right? I'm sure you've all seen this before. This is your classic example of your eukaryotic animal cell. So all of the cells that we talked about last week in tissues, like your muscle cells, the different ones, you have your epithelial cells, your blood cells, bone cells, cartilage cells, all of those different cells are all specialized. They have different amounts of the different organelles. They have different shapes. Okay, so those are all specialized for a specific function. What we're going to do today is just begin looking at the general structures that make up the cells and then looking at their functions. And the best way going forward to be able to organize this information is through a chart form, okay, where you have the list of all of the different cell parts, and then you're able to identify what the individual functions of all those different cell parts are. We're going to start off looking at the plasma membrane. Okay, so the plasma membrane is what defines the cell. It separates the extracellular environment, okay, so extra meaning outside of, from the intracellular environment where all of those cellular components are contained. And we've seen these molecules, so this should be a little bit of a review right now for us, right? Now, the plasma membrane is made up of those phospholipids, right? We have the phospholipid bilayer, where the hydrophobic fatty acid tails are going to orient themselves towards the interior, and then the phosphate heads are going to orient themselves to the hydrophilic, like the outer portion of the plasma membrane. So here you have hydrophilic heads that are facing the intracellular compartment, and here you have the polar heads that are facing the extracellular compartment. So as I mentioned, the plasma membrane forms that boundary of the cell. So what we're looking at here is sort of a zoomed in image of what you're seeing here. Okay, so the plasma membrane here is colored in sort of brown, and then here we're zooming in to see what the actual structures are that make up the bulk of that membrane. Now, because the phospholipids are amphipathic, that causes or creates an environment here within the plasma membrane that is hydrophobic. So the hydrophilic extracellular environment and the hydrophilic intracellular environment, but you have a hydrophobic environment in this plasma membrane. Well, that's going to limit what can simply just pass through. And so the cell, by creating this hydrophobic environment, is limiting or controlling the permeability of that cell to all sorts of substances. All right, so water can't simply, it can pass through, but through specific channels known as aquaporin channels. We have other channels that, which are membrane proteins that are there for a specific ion to be able to pass through into the cell and out of the cell. All right, so that's known as selective permeability. And the fact that the plasma membrane prevents certain specifically polar molecules from being able to simply move from the extracellular environment to the intracellular environment, it makes them selectively permeable and allows them to control what's coming in and what's going out. Inside the cell, we have what's known as cytoplasm. And I want you to think of it as like a jellyish kind of substance in which all of these organelles are going to be suspended in. So it sort of forms the fluid medium through which everything is suspended on the interior of that cell. Now, this structure that we have here, this phospholipid bilayer that's formed, this is known as the fluid mosaic model. And I have a video that's on the YouTube channel that you should check out that gives a bit of a dynamic perspective as to how these plasma membranes are not just rigid structures, but they're just held together by these hydrophobic versus hydrophilic interactions with the environments around them. So a bit of review here, the phospholipid is known as amphipathic, and the reason it's known as amphipathic is because it has two regions, one that is hydrophobic, the fatty acid tails, and then you have a hydrophilic region, which is the phosphate head. All right, so the hydrophilic regions are water soluble, whereas the hydrophobic regions with the nonpolar fatty acid tails, they are water insoluble. Now, we've talked about different proteins and the different functions that they have. Here we're going to focus on membrane proteins. We have four main classifications that we're going to be looking at: receptor proteins, antigens, transport proteins, and then enzymes that are embedded within the cell membrane. And the whole goal here is for these proteins to be able to assist the cell either with identification or being able to help catalyze reactions that need to take place inside the cell, or being able to allow substances to come in or substances to go out, and then also to help to catalyze reactions that are taking place within the cell. All right, so we have receptor proteins, and these are embedded within the cell membrane to be able to bind substances from outside of the cell that will then kick off a whole cascade of reactions to take place, which will eventually kick off other messengers within the cell to cause certain things to happen. Right? The one example here that I'm giving you is of non-steroidal hormones. So non-steroidal hormones, we mentioned this, I think, in tutorial, they are protein-based. They have an amino acid base to them. In non-steroidal, having an amino acid base, they are water soluble. So remember, what kind of environment do we have in that phospholipid bilayer? Right? This is a hydrophobic environment. So a hydrophilic molecule, a polar molecule, is not able to just simply pass through to enter into the cell. So instead, we have what's known as a first and second messenger system. So the hormone, the non-steroidal hormone, is going to come and bind to the receptor protein that's embedded in the cell membrane. And that's going to kick off a whole series of reactions that's ultimately going to activate a second messenger within the cell to then tell the cell's nucleus what to do. Other proteins act as markers. So antigens are examples of protein. Think of them as almost like flags. It's your cell's way of identifying, hey, I am this, okay? So we have self-antigens, which our immune system can recognize that they are supposed to be there. Now, in contrast, there are antigens on all cells, right? Not just our own cells. So any foreign pathogen, like a bacterial cell, is also going to have its own antigens on the surface of that cell. And that's going to help to also identify to our immune system, hey, that's not supposed to be there, and we need to take care of that. So antigens in general, they act as markers. Now, here's one example of blood proteins that exist on the surface of our red blood cells. This is what determines whether you are type A, type B, type AB, or type O. Okay, and just to go over this quickly, we have A antigens and B antigens. Those are the only two different types, but the organization of them dictates one of the four ABO blood types. So if you have an A antigen on the surface of your red blood cell, you're type A blood. All right, if you have a B antigen on the surface of your red blood cell, you're type B blood. Now, if you have both A and B antigens on the surface of your red blood cell, then you're type AB. Now, you are the universal recipient. And in this case, if you have no antigens on the surface of your red blood cell, you are type O, okay, which is known as the universal donor. Now, I do believe we get into this a little bit later when we look at the immune system and how it functions. So I'll leave it at that for now. But your antigens, think of those as sort of markers or flags on the cell that can help your own immune system be able to identify that cell as self or as something that's foreign. Now we have lots of different types of transport proteins. We're going to take a look at sodium-potassium pumps when we're looking at movement across membranes. We're going to look at ion channels. We have voltage-gated channels that open when a certain voltage is reached, and that's what actually stimulates nerve conduction. We have other ion channels that allow for ions to pass in and out, both through active and passive processes. All right, and we'll go through these terms, active versus passive, when we look at cell transport next week. And then last but not least, you can also have enzymes that are embedded within the cell membrane. And enzymes, remember, help to catalyze reactions. So if you're wondering if you're looking at a term and you're wondering if it's an enzyme, you're looking for the ending ASE, right? That's going to tell you that you're looking at an enzyme. And enzymes can help to catalyze those two types of reactions, both the dehydration synthesis reactions as well as the hydrolysis reactions. Now, as we mentioned, there are two major types of cells. We've already talked about this in our first lecture, actually, in week 1. But now we're going to take another look just to review this here. Okay, so we have prokaryotic cells, which are simple, and we have eukaryotic cells, which are more complex. So here's your image to help you remember prokaryotic versus eukaryotic. Okay, so a couple of things separate them from one another. First off, your prokaryotic cells lack a nucleus. So here in the eukaryotic cell, we have a membrane-bound nucleus, right? And inside the nucleus, that's where all of our DNA is stored. With the prokaryotic cells, an example of one is a bacterial cell. Okay, they just have what's known as a nucleoid region, and you can see that labeled and identified there. Okay, that nucleoid region is where their DNA just tends to be condensed towards the central portion of that cell. Okay, so prokaryotic cells lack a nucleus, whereas eukaryotic cells possess a nucleus. Right? Prokaryotic cells do not have an extensive system of internal membranes, Whereas the eukaryotic cells do. They have internal membrane-bound compartments, specifically your membrane-bound organelles. Okay, we're going to go through each of these organelles that's located in the cell, and you're going to know each of their functions by the end of today. So some examples of prokaryotic cells would be eubacteria and archaeobacteria, and then all other organisms other than bacteria possess this eukaryotic cell type. All right, so let's take a bit of a deep dive here into looking at prokaryotic cells before we move on to looking at eukaryotic cells for the rest of today. Now, prokaryotic cells are the simplest cellular organisms. Okay? They have a plasma membrane, which we do in our eukaryotic cells as well. And you can see that identified here. That's the internal layer. Something that they have in addition, which we don't have in our animal cells or typical eukaryotic cells, are the additional outer layers here. So they have a cell wall, which you only find in plant cells. And then outside of that, they also have a protein capsule that surrounds them. Now, the cell wall is made up of carbohydrates, which give a sort of a rigid cell structure to them. Okay, now in plant cells, this is the polysaccharide cellulose. And then the capsule around them is made up of proteins. Okay, so similar to how a virus is made up of a protein coat or a protein capsule, your prokaryotic cells also are, think of them as almost like sealed in a protein capsule. Now, the interior of the prokaryotic cell shows very simple organization, right? So we have that plasma membrane is the inner membrane in this case. And then inside, they have cytoplasm, as do eukaryotic cells. But there's little or no internal support framework. Okay, when we get to looking next week at all of the different proteins that create the cytoskeleton inside of our cells, you're going to see it's a much more complex structure in the eukaryotic cell versus the prokaryotic. They also have ribosomes. So this is something, when we learn the organelles that make up the eukaryotic cells, ribosomes are what make proteins in the eukaryotic cell. Prokaryotes also contain ribosomes. But this still fits with the fact that they have no membrane-bound organelles because ribosomes are not membrane-bound organelles. All right, so similar to eukaryotic cells, they are capable of producing proteins from their DNA. And then as we mentioned before, they have that nucleoid region towards the center of their cell, and this is where the DNA is localized. Now, this allows them to then divide very rapidly because they don't have to break down the nuclear envelope to allow for the DNA to be able to split between two cells when they divide. Because everything is just floating in the cytoplasm, they can simply double up their DNA and then split into two new cells. And this is why bacteria are capable of reproducing so quickly. A couple of external structures that are found on prokaryotic cells. We have the flagellum. We've talked about that already. The flagella is that tail-like structure that we have on the spermatozoa in humans. The flagellum is utilized for locomotion to allow them to be able to move around within their environments. Now, these might look like cilia, right? They might look like little tiny hairs that are going to help for movement and detection. Cilia are different from these structures. An individual one is known as a pilus, and collectively the plural is pili. All right, so these pili are there to actually help them be able to attach to other substrates that they are going to then either ingest or to exchange information with other bacterial cells. So here's where this comes into play. When a bacterial cell is going to go through and divide, and all they have to do is duplicate the DNA, and then they split the cell into two, and now you've got two daughter cells that were identical to that parent cell. So if they weren't able to exchange genetic information with the external environment or with other cells, they would all be identical and they wouldn't be able to create the genetic variability that would be able to, ward off our immune systems and trick us. So what they have is the ability to be able to, through conjugation, transfer DNA from one cell to the other. And then they can integrate that new DNA into their own. Okay, so now they've increased their genetic variability that way. Okay, so we're going to focus the rest of our time together looking at eukaryotic cells. Now, if you haven't at this point, I would highly recommend taking a look. There's a couple of videos. First off, there's a crash course video called, I think it's Eukaryopolis, right? Where Hank takes a deep dive into everything that we're going to be talking about with our eukaryotic cells going forward. And then there's another one that gives you sort of more of a generalized, but it's a really good animation and visual that shows you, it takes you through each of the individual organelles, and it talks about their individual functions. All right, so one of the things that differentiates eukaryotic cells from prokaryotic cells is the fact that eukaryotic cells contain membrane-bound organelles. Now, eukaryotic cells are much larger. They are much more complex than prokaryotic cells, as we've mentioned before. They have that plasma membrane that encases the cytoplasm. Remember, this plasma membrane is made up of the phospholipid bilayer. And then they have the internal membranes that form all of those different compartments known as the organelles. Now, the cytoplasm, it's the semi-fluid. So I just said, think of it almost as like if you've ever had that Jell-O salad, where you sort of add a fruit cocktail into it with the Jell-O. All right, think of that as your cell. Now, it's a lot more dynamic. In the Jell-O salad, nothing's moving, right? It's all staying still. But within here, this is almost like a jelly-like medium where everything is capable of moving around through the cell. All right, and the way that they do this and the way that this is sort of supported is further by the cytoskeleton. All right, next week we're going to talk about the cytoskeleton in a lot of detail. We're going to look at the scaffolding that helps to keep the cell structure. All right, so we're going to start off here with our control center of the cell. This is our nucleus. All right, so the nucleus is a double membrane-bound organelle, which acts as a compartment for our DNA. So all of our DNA is stored within the nucleus in the form of chromatin. You can see that listed here. Now, chromatin is essentially all of our DNA like a bowl of spaghetti. Okay, so it's not condensed into specific chromosomes yet. That only happens when the cell is wanting to divide. The rest of the time through the cell cycle, all of the DNA is going to be in that form of chromatin within the nucleus. So the DNA is where we're storing that hereditary information. We have the surface of the nucleus, which is bound by the double membrane. This is known as the nuclear envelope. And you'll notice here that the nuclear envelope has a number of pores. Right? These nuclear pore complexes are what allow the messenger RNA transcript to be able to leave the nucleus. Once the copy of your DNA has been made, that mRNA, messenger RNA, is able to leave the nucleus and go out into the cytoplasm, where it then can be translated into a protein by a ribosome. All right, so here in the nucleus, we have our DNA, and at any point in time, if we need to make a specific protein, we then take a copy of a portion of that DNA known as a gene. And when you take that copy, that RNA is going to leave the nucleus through the nuclear pore complex, exit the nucleus to then be in the intracellular space in the cytoplasm. And there it's going to be translated and made into a protein. Now, remember, in its uncondensed form, our DNA is known as chromatin. Now, when our cells are wanting to divide, that nuclear envelope breaks down, and all of that chromatin becomes condensed into what is known as our chromosomes. All right, so we're taking just all of the spaghetti DNA, and we're taking it then, and we're winding all of that DNA around these proteins that are known as histones to then become organized. You can see how this organization is happening into these structures here that are known as chromosomes. When we get to looking at cell division in chapter 5, we're going to be looking in a lot more detail, all of the different stages of the cell cycle. We're going to look at the control points and how all of this is regulated. So for now, as long as you've got a general appreciation for it, that's sufficient for this point in the course. All right, so the DNA is loosely coiled into strands called chromatin. Okay, that's why I'm telling you to think of it as like a bowl of spaghetti. And during the cell cycle, when a protein needs to be formed, we're going to be taking that RNA copy or making that RNA copy when the DNA is in this uncondensed form. All right, so we're going to be in the chromosomal form when the cell is going through cell division. And then when we're actually going to be the rest of the cell cycle, we're going to be taking copies of that DNA to then produce proteins. It will be in its chromatin form. All right, now you might have noticed that right within the central portion of the nucleus, there's this darkish kind of region. That's known as the nucleolus. So the nucleolus is the site for the subunits of the ribosome to be created. So the nucleolus is where you have ribosomal RNA. All right, and the ribosomal RNA is what actually makes up those subunits. So this is a larger scale image of what the ribosome looks like. You have two subunits, a large subunit and a small subunit. And the ribosome is responsible for essentially translating that genetic code, the mRNA, into a protein, a polypeptide. All right, so the RNA responsible for making these ribosomes is stored here in the nucleolus. And then when we need them, the ribosomal RNA that forms the subunits is able to assemble those. And then those subunits are capable of leaving through those nuclear pores. And then that final ribosome can be assembled in the cytoplasm. Okay, so you have all of your ribosomal RNA is stored within the nucleolus. That ribosomal RNA can then assemble into those individual subunits. They leave the nuclear pore complexes into the cytoplasm, and it's in the cytoplasm that they're put together, large and small, to make your functioning ribosomes. All right, so differences here, the nucleus, right, is this whole structure, and the nucleolus is this small region right in the center of the nucleus where the ribosomal RNA is stored. Now, extending directly off of the nucleus, okay, from these nuclear pores, they feed directly into the network known as the endoplasmic reticulum. Now, we have two different types. We have the rough endoplasmic reticulum and we have smooth endoplasmic reticulum. Okay, and what the job of the endoplasmic reticulum is to produce macromolecules. So we've already seen the ribosomes, right? They were red when we looked at the picture before. So all these little dots you're seeing, these are all ribosomes that are part of the rough endoplasmic reticulum. And that's why it's referred to as rough. Okay, so if we're looking at this image here, the rough endoplasmic reticulum is structurally located closest to the nucleus because that's where the mRNA can then leave the nucleus to then go be translated in a protein formed from it. So then we also have the smooth endoplasmic reticulum. And the smooth endoplasmic reticulum is responsible for the production of fat as well as carbohydrates. So really, you have your nucleic acids that are stored in the nucleus, right? Your DNA, RNA. And then your proteins are made by the rough endoplasmic reticulum. And then the smooth endoplasmic reticulum is what's making fat as well as carbohydrates. Now, the endoplasmic reticulum is an extensive network of internal membranes, right? So we have the rough endoplasmic reticulum, which essentially is connected pretty much directly or immediately to the nucleus, and then the smooth endoplasmic reticulum. Now, some of the membranes form channels and interconnections with one another. And then there's other portions that can become isolated. And so once you have your protein created, that these different parts of the endoplasmic reticulum can actually kind of bubble out and then form its own structure that's freely floating within the cytoplasm known as a vesicle. Right? And those are the transport structures. That's how we take something from point A to point B in the cell. Now, the segment of the endoplasmic reticulum dedicated to protein synthesis, that is the rough endoplasmic reticulum. Okay, and it looks rough because you have all of the ribosomes that are attached to the surface of that membrane-bound organelle. Now, the smooth endoplasmic reticulum, as I mentioned, is the portion that's going to be manufacturing those carbohydrates as well as lipids. So you may see in certain parts of this text or other texts that it might refer to the smooth endoplasmic reticulum as making cell membrane parts. Well, let's think about that. What is the cell membrane made out of? What's that plasma membrane made out of? Phospholipids, right? And then we have other proteins that have carbohydrates attached to them. So we have both lipids, the phospholipids that make up the membrane, as well as the carbs. So if you see that where it says the smooth endoplasmic reticulum function is making membrane parts, that's what it's referring to. It's making carbs, it's making fats. Now, what we can also do is we can then modify the proteins, right? So remember, either a free-floating ribosome or one bound to the rough endoplasmic reticulum is going to be producing the protein from the messenger RNA. Once that protein is made, it then passes off in a vesicle. So a part of that membrane pinches off, and it's essentially, think of it as like a bubble that comes out of that. If you look at the one video that I have there about inner life of the cell, you'll see what that looks like in sort of a dynamic real time. All right, so that's the way that we can transport the protein made in the rough endoplasmic reticulum to the modifying organelle. All right, and that's known as the Golgi complex or Golgi apparatus. All right, so once we have made that protein, it passes along in a vesicle, transport vesicle, and then that vesicle is going to fuse with the membrane of the Golgi. And you have two different sides, one where you have the incoming protein, and then one where you have the outgoing, whatever it is, modified protein. So that incoming protein comes in, it enters into the Golgi complex, and this is the organelle where any modifications are going to be made. All right, so remember those fats and those carbohydrates that the smooth endoplasmic reticulum is making? Those are also transported to the Golgi, where they then can be added to your protein, and you can create then a glycoprotein or a lipoprotein. So the main function of the Golgi is to collect, package, and then distribute that final molecule. Okay, one example we've talked about is the proteins coming from the rough endoplasmic reticulum being modified and then sent out. Okay, so whether from this point the secretory vesicle is then going to fuse with the plasma membrane of the cell and send that protein out of the cell wherever it needs to go, or it may be creating a protein that needs to be used in the cell, so it'll stay in and be used for its purpose. It's pretty remarkable when you think that this is going on in every single one of our cells, and they are just simply capable of knowing when to divide, and as many as we have, for the most part, they do their job faithfully throughout our lives. So there are other organelles within the eukaryotic cells that also contain their own DNA, right? And so the idea here is that these organelles appear to have been derived from ancient bacteria that were then assimilated by the eukaryotic cell, and then the eukaryotic cell evolved to then use that as a certain structure or certain part within the cell. So within all of the plant and animal cells, we have what are known as the mitochondria. And the mitochondria, those are the powerhouses of the cell, and we're going to look at those in a lot of detail in chapter 4. They're responsible for taking our glucose and being able to take the products of glycolysis through pyruvate oxidation, through the Krebs cycle and electron transport to produce ATP, which is our usable energy. All right, so both plant and animal cells have mitochondria. The chloroplasts, however, are only found in plant cells. Okay, but remember, both of these structures are cell-like in the sense that they also contain their own DNA. Mitochondria, as I mentioned, are the cellular powerhouses, right? Through oxidative metabolism. So oxidative meaning we require oxygen for the metabolic process of cellular respiration to take place. So this is where that one glucose that's gone through glycolysis outside of the mitochondria, its product is going to be brought in, and then you're going to go through the Krebs cycle and the electron transport chain, producing tons of energy for that cell. Now, the mitochondria has sort of a double membranous structure. There are inner membrane compartments and then an outer membrane that houses it all together. And between those two, we have what's known as an intermembrane space. So let's go through some prefixes here. Extracellular, right, refers to outside of the cell. And then intracellular was inside the cell, right? So intra means inside or within, whereas inter means between. So in this case here, intermembrane means it's the space between the two membranes, your inner membrane and your outer membrane. Okay, and then deep within the compartments of the inner membrane, that's where we have a structure known as the matrix, the inner matrix of the mitochondria. Mitochondria share many similarities with prokaryotic organisms, and this is where that theory of assimilation came from. They're approximately the same size, about 1 to 5 millimeters. They have small circular DNA molecules that self-replicate. We're going to talk about these small circular DNA molecules, which are known as plasmids in bacterial cells, as we progress through the course. Mitochondria replicate by binary fission. That's the same process that bacteria use. Okay, that's that process by which the DNA gets duplicated and then it splits into identical copies. And then also mitochondria have their own ribosomes, and they're similar to bacterial ribosomes. So what that means is the mitochondria have their own DNA, and from their own circular DNA molecule, they are able to produce proteins to be able to function. So we've already seen these guys before, and we're going to spend a lot of time with them once we get to the winter term when we look at DNA. The ribosomes, think ribs, all right? Ribosomes are there for protein formation, all right? They're the protein factories of the cell. They're made up of rRNA, which stands for ribosomal RNA. And remember that ribosomal RNA is stored within the nucleolus, in the central region of the nucleus. The subunits are assembled as needed. They leave through the nuclear pores. And then in the cytoplasm, that's where the large subunit and the small subunit are then put together. All right, and so what happens is, once they are assembled and put together, they are ready to then read that messenger RNA molecule. And it's almost like it feeds in kind of like a shredder. If you have a paper shredder, you put that piece of paper and it just feeds right through it. Picture that. So if you're to feed in that mRNA, single-stranded mRNA, it's going to read that 3 bases at a time, and each 3 bases, it's going to add a different amino acid. And then you're going to end up with a long chain of amino acids, which is our polypeptide. Now, one of the keys in the definition here is the ribosomes are not membrane-bound organelles, and that's why our bacterial cells can possess them. All right, the last couple that we're going to take a look at here, the lysosomes, which I want you to think of almost as like the garbage collectors of the cell. The Golgi complex has the ability to not only produce those secretory vesicles, but also produce these lysosomes. So they're membrane-bound structures that have specific enzymes in them, and those enzymes allow them to be able to break down either worn-out cell parts, right? So through hydrolysis, they're able to break down into smaller macromolecules and monomers. And then those components can be recycled to form new cell parts. And they are also capable of engulfing and digesting any particles that may have come into the cell that are not needed to be there. They're able to go around kind of and picking up the pieces, picking up the garbage. So think of them as that. Think of them as the garbage collectors of the cell. So the last organelle that we're going to take a look at here are their peroxisomes. One of the main functions is that they are capable of breaking down your larger long-chain fatty acids, right? And remember, it's not just glucose that we can produce ATP from. We're also able to break down our fats as well as our proteins. Proteins are not ideal because they produce nitrogenous waste. So the second go-to is to break down fats. Well, the peroxisomes are capable of breaking down long-chain fatty acids, sending them off to the mitochondria to then produce ATP from their breakdown. They can also then break down fatty acids within our neurons to help produce myelin. And myelin is the insulation that surrounds our neurons that have to transmit signals down long passages. So example, your motor neuron that's transmitting from your spinal cord all the way out to a muscle, it needs to be myelinated to make sure that that signal can get there as quick as possible. And so the peroxisomes in those cells are capable of breaking down the long-chain fatty acids so that they can produce the myelin that's needed to insulate the neuron and help with the propagation of that signal. Now, another major function here of peroxisomes is their ability to break down toxic substances. All right, so one of the main roles of the liver is to act as a filter for us, right? And so specifically the liver cells, the hepatocytes, they're capable of breaking down alcohol and formaldehyde. And as I mentioned before, certain types of specialized cells are going to contain more types of a certain organelle than others. Like our cardiac muscle cells, they're going to have far more mitochondria than, say, our skeletal muscle cells. And that's because the cardiac muscle cells are always having to contract, right? They're always working. And so they have evolved to contain, and so they contain more mitochondria than the skeletal muscle cells do. In the same way, liver cells, the hepatocytes, they have an abundance of these peroxisomes, right? Because that's the main job of the liver. The main job of the liver is to be able to break down and metabolize alcohol. And so we need a whole bunch of these peroxisomes in the cells of the liver to be able to carry out that function. Now, the last organelle that we're going to take a look at today that's going to come into play a lot more when we look at cellular division are the centrioles. Now, the centrioles are found in most eukaryotic cells except for plants and fungi. They're involved in our animal cells in the organization of structures known as spindle fibers. Okay, next week we're going to look at the different types of intracellular proteins, and one of the types are microtubules. And microtubules are responsible for transport within the cell, just generally speaking for now. And one of the roles that they play, specifically as spindle fibers, is that they are, during cell division, going to be attaching to the chromosomes from either side and helping to pull them apart into opposite ends of the cell. So you can see the centrioles there blown up. The centrioles during cell division are going to then separate to opposite poles of the cell so that each centriole is going to be pulling the chromosomes that have lined up in the center to opposite poles. And we're going to be going through that a lot more detail once we get to that part of the course. So for now, just understand that they're involved in the organization of those or creation of those spindle fibers. And the whole goal is to be able to transport those chromosomes into the opposite end of the cell when the cell's dividing. Okay, and that brings us to the end of today's lecture. Next week, we're going to be focusing on chapter 3.5 as well as 3.6. We're going to look at the intracellular and extracellular proteins, and then finish off our discussion on cells, looking at how cells are capable of moving substances across their membranes. Now, if you have any questions before we meet for tutorial, please feel free to send me an email. Otherwise, I look forward to seeing you all in tutorial. Take care.